Okay, good morning again, everyone. Today we are going to continue on microarray data analysis, and I'm going to show you more examples, and I'm going to show different data. We have seen how we can download microarray data from NCBI Gene Expression Omnibus, the NCBI GEO. I have found another data set that I wanted to share with you, and I'm going to show how this data set can be visualized, statistical analysis on it may be performed, and how it can be clustered using certain methods. So microarray data is extremely high dimensional because we have many transcripts that may be active at a certain time in a cell. So it's on the order of tens of thousands. So we have a lot of genes, transcripts. And the experiments, on the other hand, those chips that we use to get this data, it usually, since they cost a lot, we cannot have many samples. The experiments we perform usually on the order of tens or maybe at most hundreds. For example, let's take a look at this data set that I found again by browsing through the human gene chip that we did last time. So it was this one, this gene chip, which was HGU133 plus 2, F matrix human genome U133 plus 2.0 array. So this was an old array, but many experiments have been performed on it. There are 5,000 experiments performed on it. And when I clicked on this more, I have these experiment names, and I just selected one of them, which was genome-wide profiling of airway epithelial cells in asthmatics, smokers, and healthy controls. So there are samples taken from airway epithelial cells from people with asthma, smokers, and they're also healthy controls. So it's a comparison of many different groups. The overall design was comparing people with asthma to healthy controls, smokers to healthy controls, and then there was also in people with asthma, they were trying to maybe measure the effect of some drug for asthma, this fluticasone. I don't know what this is. And there's also the same group, another group with asthma, this time treated with placebo. Okay, so they also tried to see the effect of the drug. So we can, as you can see, we can conduct experiments, and we can design a set of chips, microarray chips, design an experiment, collect samples to compare most of the time between two cases. Samples from people with asthma or healthy people, and smoker versus healthy, or drug treated versus placebo treated as the control group. So this data set contained, as you can see, 118 samples. Each of these samples, they correspond to one of these chips. Okay, so let me show you that again. So they use 128 of them. 118 of these chips, and they are grouped. For example, these are sample IDs here, which doesn't mean anything to us right now. Which ones are healthy controls? Which ones are maybe they may have certain. I think this is for smoker, maybe healthy. I don't know. I mean, there's some patient ID that the designers of the experiment gave themselves, so these IDs may mean nothing to us. So we need to look at some metadata that tells us what these samples are. And actually, that's metadata is most of the time contained in this series matrix file. I downloaded this series matrix file, and let me show you. So by the way, I'm recording here. So this is that series matrix file. After you click on this link, series matrix file, it gets you to this one. Gets the same. I already visited that page. A 33 megabyte zipped tab separated file. So last time we opened it in Notepad++, which didn't, which we were able to browse. We were not able to browse it effectively. But you can open this after you unzip this zip file. You can open it with Excel, a spreadsheet program which can read, or any spreadsheet program which can read tab-separated files. So after we open this, let me zoom in. Let me make this a little bit smaller so it fits on my screen. Okay, and also let's zoom in on this. So there are these are the metadata. It describes different properties about the data. What's, again, some of these description that we had on NCBI Geo website. It's also here. And the actual data starts somewhere here. These are still sample information. So sample title, sample geo accession. So these are some metadata about the samples that are one of those 118 samples. So when was it public, when was it collected, or things like that. The type of the sample, it's RNA. Channel count, it's a single channel experiment, which means that on a single chip, we get measurement for only one type of sample. Last time I talked about this competitive hybridization, where we put samples from two different RNA from two different samples, and they competitively hybridize on the same chip. So this is, as you can see, channel count here is 1. This matrix chips are single channel chips. I mean, this type of chip at least. So what kind of sample source name? How did they collect the sample? Airway epithelial brushing. It's the sample is from Homo sapiens, and sample type asthmatic as baseline. Yeah, this is, so here are, this is the characteristic metadata is what we're looking for, actually, the sample type. All the others, total RNA, unknown, the protocols that they use, total RNA, chip type, address. So these are part of the address. And here, the actual data, after we see the series matrix table begin, this is the part where the actual data starts. So the rest of this data, as you can see, the first column is transcript prop set IDs, which can be converted to gene names. So these, we can get the gene names if you search for a transcript ID, or you can also find the gene names associated with these transcript IDs if you go and look at the metadata related to that platform, the chip information. The way to get that chip information is to go back to this in NCBI Geo, go back to the platform website for this chip. And here there should be an explanation about what they are. Yeah, look at this data table. As you can see, these are the IDs we have only on that series matrix file, but it contains, for example, gene bank accession ID. So which gene this prop set corresponds to could be found by looking at this gene bank accession numbers. There are, when you say download full table, for example, total number of 54,000. There's information on this platform about 54,000 transcript prop sets. So if we download that, let's see, oh, it's like 100 megabyte file. So this is the metadata about that chip, that if you want to, the annotation about those, but yeah, it's here. So this one. So we don't have samples anymore. So this is information about an F matrix chip. And as you can see, these are the transcript IDs we have in our series matrix file, but each transcript has lots of other information. It's gene bank accession ID, RefSec ID, again gene bank accession, some metadata saying what is this gene in terms of its gene ontology annotations. For example, it's a protein binding gene. So these are also annotations. Where is it localized? Gene ontology is a biological term, uh, ontology, which gives you standardized keywords about the molecular function, biological process, or subcellular localization of those genes. And as you can see, this one is, for example, for this gene. If you look at this transcript, you can say, oh, it's localized in the membrane, and this is what biological process it takes part in. Integrin mediated signaling pathway, for example. So, okay, so we have this information for all these transcripts. So afterwards, for example, if you find that certain transcripts are differentially expressed, you can go ahead and come back to this annotation data. Many tools actually load annotation automatically, but these transcript IDs that may not mean anything in the series matrix file, they have a lot more information. If you go ahead and look at the platform file, the gene name, these are the gene canonical, the gene names here, and lots of information about these genes. As I said, its molecular function, its localization, and which biological process it takes part in. So this is an important file too. But the experiment result file that we have here is this, and these numbers that you see here shows the amount of expression of each of these transcripts. And the amount of the expression, as you can see, they're just floating point numbers. They are normalized intensities. They are normalized between samples, probably, or some normalization technique is used so that they become comparable against samples within the sample too. Okay, so there may be this normalization is done to reduce variation that may be due to some technical variation between different chips or different samples. So this normalization helps us to have a way to be able to compare the transcript expression between them. So here is out of all these different types of data, the one that is important is the sample type. Asthmatic as baseline, or after fluvalent, after placebo. So this is an asthmatic sample, which was the gene expression was found after it was treated with placebo. This was after it was treated with Fluent. So Fluent, I think, is a known asthma medicine. And if you look at this part, there's another baseline, how many samples of type B have. There is less of osmotic samples, and you have healthy controls. So this is this column corresponds to a healthy control, and healthy control, and we should also have smokers. Yeah, and this column corresponds to a smoker. This column corresponds to another smoker. So we need to, as you can see, each of these samples have some labels. They belong to smoker, asthmatic, baseline, after fluent, after placebo. So this 53rd row is something that is important for us. Let's keep that. And I'm going to just delete the other parts that I'm not going to use to clear it up a little bit. Okay, so I'm going to delete the first 52 rows as well. So this is then becomes it contains all the information that I need. I have the sample types, I have the GSM ID, and also if you don't need the GSM IDs, you can actually even delete those two, but I'm going to save this again as a top separated file. Now I'm going to show you how you can read this file by a tool like MEV. So I have an old version of this TM4 MEV, but there are really many libraries that you can use on R, Bioconductor, or in Python there is BioPython. So you'll find many packages that can read such kind of a series matrix file and analyze the expression data. So I've now executed an old version of this multiple array viewer tool that I have on my desktop. The newer version uses some other new technologies to install, like Docker. They just provided as a Docker image, so you have to install Docker on your machine. So I didn't want to do that, so I just had these Java-based previous version on my machine, and therefore I'm showing it on this old version. So let's load this data from our tab separated text file. It was the series matrix, right? So let's see whether it's the correct one. Yeah, we have the sample type, the ID ref. So there are two rows of column descriptions. So it says click the upper leftmost expression value. This is the expression value, upper leftmost expression value. It's a single color array. I don't want to load annotation at this point. When you say load, the whole matrix is red, the values. As you can see now, I have nicely, since I kept that line, which one was arithmetic at baseline or the sample types are shown to me nicely. The first row is used as a label, and I can see the sample labels are nicely shown to me here. And the data itself is this one. It's color coded with respect to expression. Low expression is indicated by a greenish color, and high expression is indicated by a reddish color. All right, so I have each row you see here corresponds to a transcript, and as you can see, transcripts behave very differently. Look at let me try to enlarge this a little bit. So some transcripts are expressed a lot, and they are expressed in, for example, in all the different types of cells, regardless of their type, their sample type. So it means that these are, for example, all green ones here are probably not expressed that much. I mean, they're expressed, all green ones are not expressed, all red ones are expressed throughout the samples, but we are looking for differential expression. What is different between, for example, taking fluolant and baseline? So what is the effect of fluolant? So let's, in order to do that, we can do several things. First of all, I'm going to show you some clustering algorithms today. But we can try to cluster genes. For example, if you look at gene clusters, there is also some. These are not the clusters themselves. These are like, oh, this is some sample. I think these are, these are the expression values are also shown in terms of a signal like this, not color coded, but some. For example, there will be some different differential expression here. Let me actually try to this is the original data, gene table. You can also look at your transcripts, how they are transcribed across samples as a line graph like this. Let's conduct some analysis. For example, let's try to find which genes are differentially expressed between certain two sample types. We can do it. There are many methods implemented in this tool, like there is significance analysis for microarray SAM, which is used a lot. You can conduct ANOVA, simple Ttest. There are non-parametric non-parametric tests. I will use just simplest Ttest here. Between subjects, it's not one class. Now I can select my subjects here. For example, if you want to do between two groups, first of all, I mean, all these 118 things, you have to specify your groups. For example, if you want to compare fluolant versus placebo, or let's compare fluolant with respect to baseline. No, baseline means no treatment. Okay, so I'm going to put flavolence in group B, and all the other things, the placebo baseline is going to stay in group A, and flavolent is going to be in group B. And placebo and any other thing is going to be neither group. So first we need to label our groups. You can also do this more quicker by providing all these group information in a file, and you can actually when you say save grouping, you can see what format of file it expects, and you can play with that. You can modify that file to load grouping quickly. Let me quickly just go over them. There are 118 of them, but let's see. Baseline stays here. Flavolent goes to the group B. Let's do that. So right now, baseline, flavolent, group B, placebo here. Baseline, baseline, flavolent, baseline. I may be doing some errors here. That's okay. A couple of errors probably can be considered as like sampling error. Flavolent here. Baseline, placebo, flavolent, flavolent, placebo here, placebo here. Flavolent, flavolent, another one, placebo, this one. So baseline, flavolent. So it would be good actually if they just had these columns arranged automatically by, instead of going this way and this way, they could have arranged the sample IDs. So that all the baselines are at the first part, the flavolent treated ones come next. It will be much easier to do the grouping that way. Some samples are arranged like this, but they seem to be just randomly ordered here. I have placebo, neither group, flavolent here. Yeah, we are halfway through, almost done. Maybe I will get faster. Yeah, now we have smokers. Now I can just have them as neither group. I think the rest is going to be like neither group. Let's see. Oh, I still have baseline. Group A, healthy control. I still have some group A's that are there. Neither group, neither group, neither group. And healthy controls. And these smokers, I don't want to compare them yet. Maybe, I mean, I'm just, as I said, I just wanted to see the difference between fluent treated asthmatic cells versus the baseline, not treated with nothing. Yeah, almost done. I think the rest are all neither group. All right, so yeah, I'm done. There are different parameters for the t-test that you can set. For example, p-values based on t-distribution or some permutation testing. Are you going to use, for example, multiple testing correction, like Bonferroni correction, or you don't want any p-value correction? Do you want to construct hierarchical trees for genes? Let's do that. For significant genes, let's see how they are clustered among each other, and also whether you want to assume the two groups variances in expression are equal or not. So we are not going to assume expression is varying equally in both of those groups. They may be different, so we are going to we're not going to have assumption of equal group variance. So let's when we say okay, it's going to run the t-test, and I think it's doing yeah, gene tree, sample tree, don't want to. So, and also distance matrix selection. When you're going, this is saying if you're going to decide the distance between two transcripts in terms of their expression, what kind of distance you want to use. You can use Euclidean distance, Manhattan distance, average dot product, Pearson correlation. Pearson correlation is the most widely, most popular used one, so I'm going to keep it like at that. And when we are doing this hierarchical tree, do you want to use average linkage, complete linkage, or single linkage clustering? Let's leave it at the default options. So when I finish this, it computes everything and it's done. Okay, so we have performed the analysis. Here is the analysis the result on this pane, Ttest. Let's look at the table view, significant genes. So there are many significant genes. These are the raw p-value, adjusted p-value. Since we didn't do p-value adjustment, actually, they are the same. Can we sort this? Yeah, we can sort this by clicking on it. So there are some significant genes between these groups with really low p-value. They're really significant. We can take a look at the expression view of these significant genes, how they change over these different samples. Since all the samples are shown here, not only there were neither group, so therefore this table does not look really nice, but yeah, this expression level of these significant genes shows you how they are different in these different samples. Baseline, we also have this placebo. So all the samples are shown us together. So maybe we need to get rid of certain columns if you want to view them. But I think this expression line tells us how they are different between two groups. For example, expressed highly on the first group and not at all in the second group. So this gene, for example, it may be really differentially expressed. And you may see, if you look at these line graphs on the left, you see that there may be differences between groups, but there are other plots that you can have. How many significant genes we have? A lot, actually, like hundreds. These are, you can actually use a certain p-value threshold to actually cut these significant genes at a lower level, maybe use the top 20, top 50 of them, but that's an analysis we can perform. Let's also try to do a hierarchical clustering of all of the genes, for example, or let's try the samples actually, or both of them. When we do hierarchical clustering of all of them, so we don't have enough memory to do that. It needs more memory. So maybe we can just create sample tree. Let's see, different similar samples are going to, maybe we'll have enough memory just for the sample tree. Yeah, it's doing that. Because samples are only 118. Now we are clustering 118 samples with respect to the expression of the transcripts that they have. Now it's creating the similarity matrix, and then the clustering will be done very quickly. Yeah. So this is the hierarchical clustering. As you can see, we get ourselves a dendrogram, and similar, for example, baseline, some fluvolent is with the clustered with the baseline. So some smokers are here, healthy controls, and some other smokers. So there's the sample, actually there is a lot of variation within the samples too. Okay, so the clustering, we did not see a nice clustering of 4 or 5 separate groups. Some smokers are all over the place, so probably the data needs to be cleaned a little bit more, because it did this analysis. The hierarchical clustering was done by using all of the genes. So that's a problem. That's one of the, when I will talk about the characteristics of microarray data, that's one of the problems with, since the transcripts are on the order of tens of thousands, when we're computing the Pearson correlation coefficient between two samples, in considering them as 50,000 dimensional points, a point in a 50,000 dimensional space, the Pearson correlation between any two samples may not be very distinctive. So one thing that you may want to do is reduce these order of dimensions by, for example, removing some of those genes which do not change much between different samples. So just you may conduct this hierarchical clustering analysis on only using the top 100 significantly differential expressed genes. So which will mean that we will be reducing our dimensions from 50,000 to 100. In that case, your clustering will be much more accurate and will be meaningful. Okay, so these are the challenges working with such kind of data. So I just wanted to show how you can perform analysis and what you can cluster samples. You can cluster the genes as well based on their similarity across samples, but this is a really high dimensional nature data. So that's all I want to say actually, to give you example. That was the reason why I showed you this tool. And you can play with this and look at different statistical clustering methods that are available in these tools. And we are going to see two methods this week: the self-organizing maps and k-means clustering, which are also implemented as part of this multi-experiment, multi-array viewer tool. And some data may also be, sometimes not all the expression values are available for every sample, for every transcript. So there may be missing data. You should have methods that can also deal with missing data in some of those columns or rows. So some data may be missing. And in this microarray data, we have negative values too, because probably this is a competitive hybridization, two-channel data, in which the negative values indicate for example, under expression or upregulation. Positive values may mean upregulation, or negative values may mean downregulation. So since it's a comparison of two things, it may be a ratio of expression levels, and depending on whether if you take the logarithm of that ratio, if it's less than 1, it will be a negative value. If it's greater than 1, it will be a positive value. So positive and negative values may tell us how differentially expressed these genes are. These are yeast genes. So this is an experiment performed on yeast. And I can understand by looking at this, having negative values, it's a two-channel data comparing expression values of two samples. There are many challenges for conducting analysis on this. I told you that there are few experiments. There are many. So it says too many columns, but we have shown in our matrix that the genes were in the rows. There are too many rows when we have our genes at the rows, and samples are at the columns. So as you can see, look at this slide, it's not always a standard to have genes at the rows and samples at the columns. You may encounter different types of data in which the rows of samples and genes may have been swapped. And we may have lots of false positives due to this high dimensional nature. If you want to do certain classification, for example, I want to learn a classifier model, which by looking at the gene expression profile, try to classify whether that sample is a healthy sample or a disease sample. So we may also use microarray for diagnostics. But when you have a lot of genes to make this classification, you may overfit. In your training data, you may get really high accuracy, but because of, again, this high dimensionality, it's prone to get overfitting during training, and you also need to deal with that. So the first step usually is to reduce the dimensionality to a smaller number. There are also methods to do that. If you look at this, data reduction, there's a section in this tool called data reduction, and its purpose is specifically to do this dimensionality reduction. And one of the methods that is used a lot is principal component analysis. There's relevance networks, correspondence analysis, expression terrain map. So nice, interesting methods are implemented. But if you do principal component analysis and your goal, the cluster genes, centering mode, median, mean, none. So I just leave it at default parameters. Let's see what it's doing. So it computes the singular value decomposition of the entire matrix. So it may take a lot of time actually. They're imputing missing values. So how do you handle missing values here? Imputing means that you, for example, may assign the average expression to those missing values, or you may have some model for that transcript that you employ. But this PCA has to have all the entries none missing. Therefore, since this doesn't tolerate, PCA cannot tolerate missing values, we need to, so this Java 3D, it has a three-dimensional. I think the PCA, if it's more than one-dimensional, the PCA shows in three-dimensional the first three principal components or so. There are projections that we can take a look at. So this is the Java 2D projections are here. The first two principal components. So these are all over the place in those two samples, as you can see. So you can look at the principal components, the PC plots. There are lots of components. It's not only, yeah, I think these are the genes. That's why we have them. We need to do it on the samples as our clusters. Anyway, you can, principal component analysis is also one of the tools implemented in this, so you can play with it and do it in the correct way to reduce your data to a lower number of dimensions. We can remove outliers, normalize our data. So because of technical limitations, red color may not be shown as red. The entire array may tend to be closer to a shade of green, and therefore we may need to correct it to have the correct range, because if we know that this is really showing an overexpressed gene and it's like this, some part of green is in it, it looks like orange, then we may recalibrate, normalize the array so that the distribution of these expression values are now correct. So there are different, this shows how a two-channel array is normalized. And here I'm going to now come to these methods that we're going to discuss, k-means and self-organizing map. There are different questions we can ask about microarray data. We can do, as I said, for diagnostics purposes, we can ask whether a certain expression profile is indicative of a certain phenotype, whether it's disease or normal. We can also try to group genes or samples based on how similar they have. They show this transcript expression. For example, if you want to cluster genes, it means that we put genes together in the same group if they behave similar across these samples. If, for example, two genes, they are upregulated in the same samples together and downregulated in the other samples again together, it may mean that they are co-expressed, co-regulated, and they may be due to because they perform similar functions or they are related. Okay, so clustering is one of the useful methods that we apply on microarray data. There may be natural groupings in the data classes. We may identify new classes. For example, after we did the clustering, as you can see here, where was it? The hierarchical trees was here. When we did our clustering, we have seen that there are certain groupings. For example, certain baselines are really similar to each other. Let's see, fluovalent are all over the place. I mean, there are some fluovalent treated patients that are similar to each other, but there is no natural grouping between fluovalent, and the sample labelings do not correlate with our clustering that much. This may be due to the fact that we used all of the genes expression values, not before dimensional reduction, but it also shows you that certain samples are really close to each other, like these healthy controls or whoever the smoker is. He or she is in between, clustered between these healthy group. Maybe the smoker is tolerant to this smoking effects. I'm not sure, but we may see these natural sampling. Clustering the samples. We may see natural groupings within the samples. The same as with the genes too. Okay, we may see similar behaving genes. And there are different methods. We have seen, for example, hierarchical clustering, the neighbor joining method, and UPGMA method we have learned in phylogenetic tree construction are examples of hierarchical clustering methods. So I'm going to not show you another hierarchical clustering method here, but we're going to talk about self-organizing maps and k-means. And in any of these clustering techniques, one important thing, whether they're accurate clustering methods or not, whether the result looks good or not, depends highly on the distance measures. How do you measure quantitatively the difference between two samples or two genes? I told you that when we are measuring the Pearson correlation coefficient, when we compute the Pearson correlation coefficient between two samples, when we use all those 50,000 transcripts, the distance, the Euclidean distance or the Pearson correlation coefficient will lose its distinctive power, its discriminative power as the dimensionality increases. There is this notion of curse of dimensionality, which means that as the dimensionality increases after 5, 10 dimensions, the space or the objects in your space, they become equidistant almost however far away they are. On 2D, it's really, they may be nicely scattered on the plane, but in higher dimensions, the space becomes mostly empty, and the distances between different objects, they become almost equal, which means that in high dimensions, the difference between Euclidean distances or differences between Pearson correlation coefficients, they diminish. So whatever distance measure you're using, you have to be very careful about how they behave in high dimensions, whether it treats scaling in data. For example, imagine two genes behave similarly, but one of the genes is consistently lower expressed than the other gene, but their behavior is similar. They increase and decrease. They increase, decrease at same samples together. But if one gene has higher expression than the other, it's the Euclidean distance between them is going to be large because of this difference. But if you want to capture just the behavior, which means that maybe the scale factor, the expression level is not so important. I'm just interested in the direction, whether it increases or decreases. Instead of using Euclidean distance, you may want to use Pearson correlation coefficient, which actually takes care of these differences in scale. It first normalizes the expression values all to within the gene to some. So it's actually, this is called mean centering. It mean centers, subtracting the mean values and compute the vector angle has some kind of a similar effect than that, like scaling those expression values. But so which distance measure is best for a certain goal? There is no actually such the distance measure that works best in all tasks. You have to choose based on what you want to achieve, what similarity between samples or similarity between genes means to you. You have to choose some kind of a mathematical function to measure the similarity between two high-dimensional objects and get a single similarity or distance value. And after that, we have ourselves an all-to-all pairwise similarity or distance matrix, just like we had in phylogenetic tree construction. Remember the input matrix in phylogenetic tree construction. We have this example of A, B, C, D, E. which shows the similarities or distances between A, B, C, D, E in a 5 by 5 matrix. So all these clustering methods, if you want to cluster our 50,000 genes, for example, the original matrix here is 50,000 by 118. This is not a similarity matrix. This is an expression matrix. If you want to cluster those 50,000 genes, for example, we have to compute the similarity between all pairs of those 50,000 genes, which means that we have to first construct a 50,000 by 50,000 matrix in which the columns are also genes. And an entry here shows the similarity between gene I and gene J. Okay, so it's as you can see, it will be a really big matrix, 50,000 by 50,000, millions of entries in that matrix. And this is what we need to do first. After we have that all-told pairwise similarity or distance matrix, then we can use methods like k-means or self-organizing maps to make use of those pairwise distances to cluster those objects. Okay, before I start k-means clustering, I think it's a good time to have a break now. Let's have a 13-minute break until 10:40. I will stop recording now.